Alfred Hershey and Martha Chase wanted to identify the hereditary material of bacteriophages. They knew that bacteriophage T2 has two components, protein and DNA. How could they trace these molecules? Well, proteins usually contain sulfur, but nucleic acids don't. Nucleic acids always contain phosphorus, but few proteins do. They prepared 2 batches of T2. By growing E. coli on media labeled with phosphorus-32 or sulfur-35, and then growing T2 viruses on those bacteria, they obtained viruses labeled with P32, which marked their DNA, or with S35, which labeled their proteins. They infected bacteria with virus and after a few minutes tossed the mixture into a blender. The blender stripped away most of the S35, that is, most of the protein, but most of the P32, the DNA, remained with the bacteria, suggesting that the DNA is what enters the host cell. Next, they repeated the experiment but let the mixture stand until the bacteria lysed it naturally, producing a new generation of viruses containing almost none of the protein but about 1/3 of the DNA from the virus infection. The conclusion? DNA is the hereditary material of bacteriophage T2.